grand rounds at the House Clinic today. I titled my presentation Beyond Fluid Pressure, a Novel Disease Paradigm for Meniere's Disease. And what I want to share with you today is a mix of otopathology data. You might know we have a large collection of postmortem human temporal bones, including many temporal bones from patients who suffered from Meniere's disease. And most of our research is based on otopathology data. And over the years, we advanced this towards more clinical studies. And I want to share with you a mix of both to convince you, hopefully, that we're on track of establishing a novel disease concept for this mysterious disease. I have no disclosures. I want to start out by acknowledging the otopathology lab, all investigators, staff, and fellows there, as well as our fantastic Meniere's research group at Mass Eye and Ear. Especially want to point out Dr. Steve Rausch, who was for decades director of the vestibular division here at Mass Eye and Ear, and who just recently, I want to say, semi-retired and became a Californian, but we're still meeting weekly in our group. So I want to start out with this classic scheme of how we think about Meniere's disease. So we think there are various factors and they somehow act upon the inner ear, causing endolymphatic hydrops. And in the very first version of this schematic, it was believed that endolymphatic hydrops directly causes Meniere's disease, meaning the typical episodic vertigo symptoms, the fluctuating hearing loss, as well as tinnitus and oral fullness. But over the years, data came out, specifically otopathology data, that suggested that endolymphatic hydrops is most likely not in the center of this disease paradigm, that endolymphatic hydrops is more likely an epiphenomenon, another symptom that is not only associated with Meniere's disease, but also with a whole host of other inner ear disorders. So the center of this disease paradigm really is a black box now. We don't know where Meniere's disease starts on the cellular and molecular level. And this is what our research mostly is about. Nevertheless, our clinical concepts, the way we think about Meniere's disease, is still pretty much informed by this first concept. Endolymphatic hydrops is still in the center when we explain the clinical disease course and certainly when we talk about treatments. So here the schematic on the right side shows the typical disease course in Meniere's disease with time on the x-axis and inner ear function on the y-axis. So every Meniere's patient starts out with a normal inner ear, and over time this ear starts, for unknown reasons, to degenerate. And then there is this characteristic middle part where function starts to drop episodically or to fluctuate, and this is when we diagnose Meniere's disease. And when a patient passes through this phase, once the inner ear is severely degenerated, these fluctuating symptoms stop, and we end up with permanent hearing loss and balance dysfunction, or a dead ear. And we interpret these episodic symptoms with this classic concept of fluid overpressure in the ear, with fluctuating pressure waves that repeatedly damage the sensory epithelium, causing maybe ruptures in these membranes, mixing of potassium-rich endolymph with perilymph that then leads to excitatory phenomena and ultimately vertigo and hearing loss symptoms. And this concept was really derived from analogies to the eye disease glaucoma. Meniere's disease was called glaucoma of the inner ear because it was thought that in both these diseases, fluid overpressure in these organs causes these episodic symptoms. So also the therapies that were successfully applied to glaucoma were kind of adopted for Meniere's disease, from dehydrating treatments like low salt diets or diuretic treatments up to surgical treatments like endolymphatic sac decompression or endolymphatic shunt implantation, all with the idea of dehydrating the endolymph fluid space and thereby releasing symptoms. The problem is meta-analysis have shown that none of these therapies are proven to be efficient, and certainly none of these therapies influence the long-term disease course. They do not prevent progressive degeneration of a Meniere's ear. So we're still living with this hydrop-centric paradigm. And what I want to show you in this presentation here is our approach of how we started searching for, let's say, the origin of Meniere's disease in the inner ear and how we evolved our research over the last 5 to 6 years. So this slide is more an agenda slide. Again, as I said, we started our research looking at human temporal bones, histologically processed human temporal bones, and we focused our research pretty much on the endolymphatic sac, this non-sensory part of the inner ear that is lined by an epithelium filled with endolymph, which is continuous with the endolymph of the cochlea and the vestibular system. And I want to point out that in this schematic here, the endolymphatic sac is pretty much drawn in scale to the human inner ear. So the human endolymphatic sac is a pretty large organ, and this will be important down the road throughout this talk. What we did first, we did protein expression studies, and in line with previous research, we have shown that the endolymphatic sac epithelium hosts a whole range of ion transport mechanisms that are regulated by systemic hormones, foremost aldosterone, so really reiterating the endolymphatic sac's role as an interface between the closed-off, sealed-off inner ear compartment and systemic blood circulation in the body. We then looked at the endolymphatic sac in patients who suffered from Meniere's disease in a systematic histology study, and we made two interesting findings. We saw that in about 70% of all Meniere's temporal bones, the endolymphatic sac was really degenerated. And in the other 30% of temporal bones, we saw another pathology of the endolymphatic sac, which we call hypoplastic or underdevelopment. So in those years, the endolymphatic sac has never fully developed. It remained rudimentary. What's interesting is that these two pathologies have obviously different causes, etiologies, but they both affect the most distal part of the endolymphatic sac that reaches out beyond the operculum into the dura of the posterior cranial fossa. And this aldosterone-regulated ion transport system that I just mentioned is most highly expressed in this distal part. And it's missing in all Meniere's patients due to these different pathologies. What we did next is we developed CT and MRI diagnosis criteria to diagnose these two different pathologies in living Meniere's patients. The challenge here was, as you know, in a CT or an MRI scan, you can't see epithelium. We can't see the endolymphatic sac itself, but we see the bony outline. And because the bony outline around the endolymphatic sac is also hypoplastic in this group, as I will show you on the next slide, we're able to distinguish a degenerated from a hypoplastic endolymphatic sac, meaning we can now distinguish Meniere's patients with the degenerative pathology and those with a hypoplastic pathology. And we have shown in these studies that these two patient groups also differ in their clinical features, as I will show you later. So I'm going to spend one slide and talk about the radiological marker that we're using. What you see here on the left are endolymphatic sacs seen in a horizontal section plane, as you would see it in a temporal bone CT scan or a brain MRI scan. And what we're measuring is the angle that the vestibular aqueduct describes from the vestibule to the operculum in a normal temporal bone, as well as in Meniere's patients with a degenerative pathology, which also have a normal developed temporal bone. The vestibular aqueduct describes an angle. It has a hockey stick-like shape, and this angle is 100 degrees or less. In contrast, in Meniere's patients with a hypoplastic endolymphatic sac pathology, this whole bony structure also remains rudimentary, meaning the vestibular aqueduct is shorter and has a more open angle, 140-degree or more. And we showed this the first time in a histology CT correlation study. This is the quantitative data. The thing I want to point out here is that this is a really discriminating marker, meaning there is no overlap of angle values between the hypoplastic group and the degenerative group. So we can really use this angle measurement as a definitive criterion to separate hypoplastic from degenerative Meniere's patients. We call it the angular trajectory of the vestibular aqueduct, or ATVA. So we used this ATVA measure then the first time in a retrospective study, meaning we took about 100 Meniere's patients that had CT or MRI scans, and we classified them into the degenerative group and the hypoplastic group using this measure. And then we collected a whole bunch of clinical data from the available medical records. And as I just mentioned, we saw some very striking clinical differences between these two groups. The most significant or interesting ones are listed here. First of all, in the degenerative group, we saw a fairly balanced gender ratio, 50% male, female about. But in the hypoplastic group, almost all of these patients were male. This was a highly significant difference. Also, in the hypoplastic group, we saw that about 30% of those patients had bilateral disease. In fact, almost all Meniere's patients who at some point during their disease course progressed to bilateral disease belonged to the hypoplastic group. This was also the group that had a substantial family history for Meniere's disease, and those patients tended to have a more early onset of disease during life, although this was not a significant finding in this study. There are also differences in terms of treatment for these two groups. In this study that we just published, we showed that here on the right side, this is a typical Meniere's cohort. And as I just mentioned, 70 to 80% of the patients are degenerative, only 20% are hypoplastic. But when we look what Meniere's patients are eligible for a cochlear implantation, meaning who ends up with such a severe degree of hearing loss that they need rehabilitation with a CI, it's mostly hypoplastic patients. And this is not because they have worse hearing thresholds. It is, we think, because those are the patients who are affected bilaterally, and those are the patients who tend to be affected earlier in life, meaning the disease can play out longer in both ears, causing more damage, higher disease burden. So we showed these interesting correlations now that we were able to separate this whole cohort of Meniere's patients into clinically meaningful subgroups. The next question we were interested in our research was, well, if these patients all have pathologies of the endolymphatic sac, are those pathologies truly causative for Meniere's disease? Are they the cause of the disease? And we are answering this question in part in animal models right now, but we also use clinical studies. One prospective study that we did was designed as follows. We had a group of Meniere's patients that presented in our department with Meniere's disease in one ear. We did our CT scan. We saw that in the clinically affected ear, the endolymphatic sac was hypoplastic. And in the contralateral ear, which we, of course, also saw in the CT scan, we saw that the endolymphatic sac and vestibular aqueduct looked normal. And then we had a second cohort of Meniere's patients. They also presented in our department with onset of Meniere's disease in one ear. CT scan again showed a hypoplastic sac in the clinically affected ear, but also a hypoplastic sac in the second ear, which at that time point did not show any symptoms. So we followed these two cohorts now prospectively with the question, what happens to the second year? And if our hypothesis is right that endolymphatic sac pathology, specifically hypoplasia, causes Meniere's disease, we would expect that the second cohort, the one down here, progresses to bilateral disease, versus the first cohort should stay with unilateral disease. So the data is presented here on the right side with years on the x-axis. So the total observation period was 30 years, and it's so long because we included some of these patients retrospectively and observed them prospectively for 5 years. And on the y-axis is the proportion of patients with unilateral disease, meaning both of these groups start out here on the upper left corner. The first group, none of these patients turned bilateral, according to our expectations. In the second group, every single patient developed bilateral Meniere's disease. So this fully was in line with our hypothesis that this group is the one that develops bilateral disease, and it also supports our idea that the endolymphatic sac pathology that we observe in these patients might be causally linked to the disease. There's another interesting observation hidden in this curve here. As you can see, there are some patients, they turn or progress to bilateral disease within only 1, 2, or 3 years after the first year was affected. And then there are other patients, they have a much longer time interval before the disease shows up in the second year. It can be 2 decades or even longer. And we were interested in a recent study that we haven't published yet in the variability. Why do some patients progress so early and others so late? Although all of them have a hypoplastic endolymphatic sac according to our imaging, so the same pathology. And this is a question our fellow Arpen Bose addressed in a histopathology study. We looked at temporal bones in our collection from Meniere's patients with hypoplastic endolymphatic sacs. And you see 3 examples, 3 different years here on the left side, and they all show the hypoplastic endolymphatic sac, blue, in the operculum region, purple. And what's striking is that there seem to be differences in the severity of the pathology. So up here is a hypoplastic sac that looks fairly normal. It has a so-called rugous proximal portion where the epithelium is wounded, and the distal portion stretches a little bit into the dura. And on the opposite end of the spectrum is a severely hypoplastic endolymphatic sac down here, which is only a tiny epithelial bleb, less than 100 micrometer in size. So it looks like from histopathology, there is a spectrum of pathology. And what we did next is we took from all these specimens, we have a total of 14 years in our collection, serial sections, and reconstructed the entire hypoplastic endolymphatic sacs and measured the endolymphatic sac epithelium in square millimeter to get a more quantitative measure for how hypoplastic they are. And then we correlated this data with the age of Meniere's onset, which we had available from the medical records. And what we found to our surprise was a fairly good correlation, as shown here, between the size of the hypoplastic endolymphatic sac and the age of disease onset, meaning the most hypoplastic sacs, the ones down here, belong to the patients who had disease onset in their late 20s, early 30s, so early in life. And the Meniere's patients with the least hypoplastic endolymphatic sacs had a disease onset rather late in life, in their 60s, some even in their 70s. So a very clear correlation between residual endolymph epithelium and timing of disease onset. And this clinical data together really gave us confidence that endolymphatic sac pathologies contribute substantially, not only to the onset of Meniere's itself, but they also seem to influence the timing of the disease. want to make a cut here. So far, I talked about what is the effect of a hypoplastic endolymphatic sac onto the clinic. In a second line of research, we're looking now more upstream. We're interested in the question, what actually causes underdevelopment of the endolymphatic sac? And I already said this group is mostly male. They have a strong family history for Meniere's disease. They tend to have an early disease onset and they tend to have bilateral disease. So those are all factors that basically scream genetic cause, right? And this is what we set out to investigate. And we got from our medical records review a very interesting hint, because besides all these clinical features I mentioned, these patients with hypoplastic pathology had another interesting correlation. Many of them already had a genetic diagnosis, a disease called X-linked hypophosphatemia. And X-linked hypophosphatemia, or XLH, is a genetic disorder caused by mutations in the gene called FEX. It's located on the X chromosome, and it primarily causes bone and kidney problems. Bones tend to be hypoplastic and less dense. And these patients also tend to develop kidney stones and chronic kidney disease. Interestingly, when we did our first literature research, we found a couple case reports describing hearing problems in these patients. So we suggested that there is a genetic link between XLH and our subgroup of hypoplastic Meniere's patients. And the way we approached this was first from a observational epidemiological point of view. And the lead on this project really have my colleagues, Dr. Divyachari, Paula Robles-Bolivar, and Arpan Bose. So there's a little bit of math here on that slide, so I hope you bear with me. So first, we gave it some thought. XLH, as I said, is a very rare disease. Only 5 in 100,000 are affected. And Meniere's disease of the hypoplastic endotype is also a rare condition with only 150 in 100,000 affected. So in theory, or the odds ratio, the chance of someone having both diseases, if they are unrelated, just by bad luck, is very small. It's just 1 in 13 million. So it's very unlikely to find a patient like this. What we did is we recruited over the last 2 years 34 patients with XLH. This seems like a small cohort, but because this disease is so rare, those are actually all known XLH patients in the New England region, plus the German-speaking region of Switzerland. And what we found is that 6 of these patients had bilateral Meniere's disease of the hypoplastic endotype, according to our CT scans. This is the largest XLH cohort ever evaluated for auditory and vestibular symptoms. And with those 6 patients, the odds ratio we observed, 1 in almost 6, is several thousand times higher than the by random chance odds ratio. So this is a smoking gun that both diseases, XLH and Meniere's disease of the hypoplastic endotype, are causally linked. And it became more interesting when we looked for who had Meniere's disease in our cohort. Among our 34 patients, we recruited 26 female XLH patients and 8 males. From the females, none had Meniere's disease, and none had a hypoplastic endolymphatic sac on CT, which means all the 6 Meniere's patients we found were male, again, fitting into the scheme of our Meniere's hypoplastic group. And this leaves 2 male patients. One of them had no Meniere's disease, but on CT scan had bilateral hypoplasia. He actually was fairly young, in his mid-30s. So according to our data, we expect that he will develop Meniere's disease in the future. And the last male XLH patient was really interesting. He had no Meniere's disease, no hypoplasia on CT imaging, But when we did more in-depth sequencing of his PHEX gene, we actually found that he has a mosaicism for a PHEX mutation, meaning not all cells in his body carried a mutated PHEX gene. About half of the cells had a normal PHEX gene. So on the genetic level, this patient resembled more the female cohort. How do we tie all of this together? We think that in XLH patient, what we call PHEX gene dosage effect drives the Meniere phenotype. Let me explain this briefly. Female patients have 2 X chromosomes, which means half their cells in their body express a normal copy of the gene, half the cells express the mutated form. And that means on the protein level, they have 50% functional PHEX. And we think this is enough to produce a normal inner ear. This is also the case for our mosaic male patient. The 6 male patients with Meniere's disease, however, they have only 1 X chromosome, which means 100% of the cells in their body express a mutated dysfunctional PHEX gene, meaning they do not produce any functional PHEX protein, whatever PHEX does. And we think that this then leads to penetrance of a bilateral Meniere's phenotype. We're currently exploring this in a mouse model, transgenic mouse model with PHEX mutations, and we see actually very similar correlations there, meaning we think this is truly the first direct genotype-phenotype correlation in Meniere's disease. So after establishing that endolymphatic sac pathologies really seem to be involved in the pathogenesis of Meniere's disease, we were now interested in the question, well, how does this all tie in with endolymphatic hydrops, which is generally the hallmark pathology in this disease? We were interested first establishing on a more quantitative level how damaged is a Meniere's inner ear truly by the endolymphatic sac pathology. We've looked at a lot of endolymphatic sacs histologically, and as I said in the beginning, this is a rather large organ in the human inner ear, meaning it creates a huge epithelial surface that stays in contact with the endolymph fluid space and probably contributes a lot of ion and fluid homeostatic function to the ear. So what we did in serial sections, we set out to quantify how much epithelial surface does an endolymphatic sac actually contribute to a human inner ear, and what's the impact of endolymphatic sac pathology on that, again, on a quantitative level. So we basically, in serial sections, thousands of sections, measured the endolymphatic sac surface in square millimeters and related it to the epithelial surface in the cochlear and vestibular portions, first in normal inner ears. And to our surprise, the endolymphatic sac in a normal healthy adult inner ear contributes 1/3 to almost a half of all epithelial cells that line the endolymph space. So a huge cell mass. Then we did the same analysis again in Meniere's disease, in Meniere's ears with a hypoplastic endolymphatic sac. And this pathology leads to a massive loss of cells. So about 1/3 to a half of all epithelial cells are actually lost, meaning the endolymphatic sac in Meniere's disease is truly a major site of pathology. A Meniere's ear is severely injured because it lost 1/3 to a half of all epithelial cells. So with that in mind, we now turned around and looked at the cochlear and vestibular portion to the membranes that actually form hydrops. And we wanted to have a closer look at these membranes, specifically at their epithelial cells. And we're looking here at Reisner's membrane that, again, is made up of epithelial cells that face the endolymph space and mesenchymal cells that face the perilymph space. And we really wanted to test the old fluid pressure paradigm, which predicts that when hydrops forms by fluid over pressure, these membranes get passively distended, they expand, but the number of cells in such a scenario should stay the same. They just should, like a balloon that gets blown up, move apart from each other, meaning the cell spacing, the spaces between them should expand according to this model. And we tested this model of our fellows. Diana and Corey developed a deep learning image analysis model that counted cells in these hydropic rhizinous membranes from a whole range of normal and Meniere's affected ears and measured the spaces between neighboring cells. And I want to share some of the data with you. What you see here is a cross-section through the cochlea, the basal turn down here, the apical turn up here, and the same view of a Meniere's ear with endolymphatic hydrops. And the cell counts in rhizinous membrane are shown here as a heat map with relatively low cell counts in normal rhizinous membranes. But in hydropic rhizinous membranes, we see that the cell count, the number of epithelial cells, goes significantly up. The cell spacing, however, stays the same in both normal rhizinous membranes and hydropic rhizinous membranes, meaning the data shows the opposite. And this is the aggregate data from all inner ears Diana and Corey looked at. On the x-axis, we see hydrop severity, meaning normal inner ears with no hydrops are shown on the left side. And the Meniere's ears with the most severe hydrops are shown on the right side. And the y-axis show the cell counts. And what we see is an almost perfect correlation between increasing epithelium, or meaning increasing hydrops, and increasing cell counts versus the cell spacing stays the same. So conclusion from this study is we do not see any signs of fluid pressure in these membranes. What we see is massive cell proliferation. Excuse me. Massive cell proliferation. In the most hydropic membranes, the number of epithelial cells quadruples, or in some cases has even a 7-fold number of epithelial cells. How do we tie all of this together? So we know from our previous studies that a Meniere's disease ear carries significant damage or injury in the endolymphatic sac. There are a lot of epithelial cells that are being lost. And what we see in our most recent study, that in the other parts of the inner ear, we see massive epithelial proliferation going on. New epithelial cells are being added to the endolymph border. And to explain what's going on, we're going to turn to another organ. I'm picking the kidney here as an example, but basically every organ in our body that gets injured reacts to that injury with two different mechanisms. The first is called hypertrophy, meaning the remaining cells in this injured organ grow in size. They upregulate gene expression, meaning they put in more ion channels in their membranes. They produce more enzymes to compensate for a functional loss, to take over the function from the lost cells. And the second mechanism they leverage is called hyperplasia, meaning they undergo mitosis, cell division. They replace the lost cells with new cells. The problem is this is not regeneration, meaning the new tissue that is being formed by hyperplasia is qualitatively inferior. The blood supply is not sufficient, and this ends up in a vicious cycle, ultimately causing more organ damage, more organ injury, and again, driving more hypertrophy and hyperplasia. And this vicious cycle, in the example of the kidney, often leads to chronic kidney disease. And the clinical course of chronic kidney disease is shown here on the right side, where we start out in the upper left with a normal kidney, and upon injury, the kidney slowly starts to degenerate. It tries to repair the injury using hypertrophy and hyperplasia, which adds more damage and drives the whole kidney into a dead kidney, ultimately needing dialysis. When we look at the functional output of the kidney, it would be here blood levels of phosphate and calcium. They remain stable for a fairly long time. But in the middle part here, when the kidney is sufficiently degenerated and has functional loss, these parameters start to fluctuate depending on physiological challenges. This is called the early phase of chronic kidney disease. And once the kidney degenerates further, at some point, this fluctuation goes into a progressive permanent loss of these parameters. And you might remember the initial clinical scheme of Meniere's disease that I showed you. And we can literally put this over here and we see the same clinical trajectory, right? We start out with a normal ear that progressively degenerates. And in the middle part, we have fluctuating inner ear function, which we call Meniere's disease. So what we think what goes on in the inner ear mimics what's going on in the kidney in the case of chronic kidney disease. We have an initial injury to the endolymphatic sac, and we think that endolymphatic hydrops is an active compensatory response to the inner ear to this injury. New cells are being added. These epithelial membranes actively expand in an attempt to compensate for that injury, in an attempt to stabilize hearing and balance function. But because the inner ear is not designed to be hydropic, this ends up in a vicious circle called maladaptive progression, which ultimately drives the inner ear to a dead ear through this phase of unstable function that we call Meniere's disease. If you want to read more about this idea, we just published that in bioRxives. So to summarize, we argue that endolymphatic sac pathology-based endotypes exist in Meniere's disease. We have demonstrated this via distinct patient subgroups that we defined by clinical, radiological, pathological, and genetic features. And this altogether offers a framework to, we think, advance our understanding of this disease pathophysiology and also clinically guide personalized diagnosis and management. And ultimately, in our latest ongoing research, we redefine the role of endolymphatic hydrops as at least partially beneficial and away from the old fluid overpressure paradigm. And the one take home that I want to leave you with is we think that CT temporal bone subdiagnosis or endotyping of this heterogeneous cohort of Meniere's patients into groups with a degenerated or a hypoplastic sac is beneficial both for clinic and for research. In clinic, we have shown that we can successfully stratify patients for future risk of bilateral disease, and it also helps us counsel patients about their disease course and allows us to make better decisions for long-term management strategies. For example, the application of ablative treatments like IT gentamicin injections into ears when we expect the second ear to be affected. We're more conservative for that. And in our research, this endotyping approach really helps us to resolve the clinical heterogeneity of the overall cohort. We think in the future, this will be one approach to stratify study designs and improve cohort selection to get more significant results for treatment studies and so on. And we also think it will facilitate genotype-phenotype correlations and biomarker discovery, as we have shown in the example of XLH and Meniere's disease already. And thank you so much for your attention. Thank you again for the invitation.